Hi, today I'm going to discuss about the role and function of tumor suppressor genes and oncogenes with a brief overview about carcinogenesis. The fabric of life, or the secret of life as we all know, consists of multiple copies of a single basic unit, the nucleotide, which comes in four different forms: adenine, guanine, thymine and cytosine. The question is, number one, is there something divine at the heart of the cell that brings it to life? I think there is something. Well, it would take more than a man to make a cell, give it life, divide, multiply, and propagate. Question number two. How does the cell regulate duplication of its vast nuclear content, the DNA, without making a mistake? And the last question. Why and how does the cell become dysregulated and become abnormal or cancerous? When you have done this exercise or you have seen this presentation, you would have understood the mechanism of cell dysregulation. You've understood the difference between tumor suppressor gene and oncogene. And you would have also understood the process of cancer formation or carcinogenesis. I'm going to start with an example, an experiment that I did during my doctoral program. It was believed that chewing a betel nut causes cancer, so I started an experiment using Swiss albino mouse as my experimental model. I was trying to understand the process of carcinogenesis using this animal model. I treated this mouse with a certain amount of betel nut for a few days, probably 10, 20, 30 or 40 days, and then used the mice bone marrow to prepare metaphase chromosomes. Then this gave me a result which showed that treating the animal with betel nut caused a chromatid break. So that means a chemical can cause a damage in the DNA. So I didn't stop here. I went on to repeat this experiment using my own blood and checked what happens if betel nut is treated to human blood. Well, it created an aberration called a cystic chromatid exchange. I have indicated this using arrows. You can see one arm of the chromosome is stained lighter than the other, and you can see wherever there are arrow marks, you can see the arm of the chromosome has exchanged over to the next arm, or the cystochromatic changes have taken place. This is a DNA repair mechanism error. Let's look at what happens in the cell, or how the cell deals with DNA damage caused by betel nut or any other carcinogen. Imagine you have to go around on a bicycle and you take about 1 hour to complete. In case you encounter a problem with your cycle on the way, you would stop, repair, and then continue. This would have rectified the problem, and then you would have continued to complete the cycle. If you hadn't stopped, things would have gotten worse. A similar phenomenon happens in the cell. It goes through division or cell cycle, and it has to happen without errors. The cell has several checkpoints or signals where the process can be checked, and if everything is alright, the cell proceeds. These checkpoints are done by certain genes which are called as tumor suppressor genes. If any of these tumor suppressor genes stop functioning due to mutation or deletion or some other genetic alteration, the cell cycle checkpoint will not work, eventually leading the cell to become cancerous. One cell with a mistake would become two aberrant cells and would become more in number at the end of every cell division. 1 would become 2, 2 would become 4, 4 would become 8, and it would lead to an exponential increase in the aberrant cells. Now let's look at the role of gatekeepers or tumor suppressor genes in the cell. This was a hypothesis which was put forth by a scientist called as Knudsen, and it is called as the Knudsen's two-hit hypothesis. Tumor suppressor genes are genes that slow down cell division, and it does repair of DNA damage or errors during replication. If the error or the damage cannot be rectified, the tumor suppressor gene recruits the cell to take up apoptotic pathway, that is programmed cell death. Apoptosis is a process which controls cancer development. Now look at this picture. We inherit a copy of the gene, one from the father, that is a paternal gene or the paternal allele, and one from the mother, which is called the maternal allele or the maternal gene. Both are normal and function properly. They are in a homozygous condition as normal. Let's take a hypothetical condition. Let's say one of the allele that you have inherited either from the father or the mother is faulty, may be deleted or mutated. Here in this case I have shown the maternal allele to be faulty or affected, meaning it's not functioning in the normal way. If there was a tumor suppressor gene in this location which was meant to check DNA damage, then things will have a serious implication. Let's call the gene at this location as P53. P53 is one very important gene present on chromosome 17, which is responsible for checking on DNA damage, and if damage is found, it would arrest the cell, then repair the damage and continue along in the cell cycle. If this gene was damaged, there could be a serious problem. There would be a checkpoint problem. But the good news is we have two copies of the gene, one from the maternal and one from the paternal. Here only the maternal gene is affected. The gene from the father or the paternal gene compensates for the loss, and so there still would be a cell cycle arrest and repair process will take place, and then it will resume the cell cycle. Let's take another condition. If during the life of an individual the other allele, that is P53 from the father, also becomes damaged, then the checkpoint would be absent and the cell cannot stop even if there is a damage in the cell, and the cell would continue with the mistake and become cancerous. Now just to run through the whole thing one more time, let's look at tumor suppressor gene. We have two genes or two copies of the gene, one inherited from the father and one from the mother. There can be two possibilities. One, the cell divides normally and then goes on as a good cell. Suppose one of the inherited gene from the father or the mother is having a problem, that is a mutated tumor suppressor gene. You could have two possibilities. You can have one cell that is on the left side to be normal, and the other one that is there on the right side which could become a cancerous or have uncontrolled cell proliferation. This is called loss of function of a particular gene because it needs both the alleles to be inactivated before the cell can become cancerous. So it is called as loss of function of a particular tumor suppressor gene, which can lead to the formation of cancer. So this is called as a recessive gene because both the alleles have to be lost and it has to be in a recessive homozygously lost condition. That is why it is called as a loss of function, and it is also called a recessive kind of a function inside the cell. In contrast, let's look at a oncogene. The scenario for an oncogene is different. Here we have what is called as proto-oncogenes. They are present in the cell which helps the cell normally to grow. When proto-oncogenes get mutated, it becomes abnormal, or when it mutates, it gains a function and makes the cell cancerous. So this is called as gain of function. So I hope you can make out the difference. In a tumor suppressor gene, you need two hits, that is both the alleles have to be inactivated to become cancerous. that is the cell to become cancerous. But here in an oncogene, even if one proto-oncogene is mutated, it gains the function and promotes the cell to become cancerous. So it is called as gain of function. In a tumor suppressor gene, it's loss of function. In an oncogene, it is gain of function. And it is also called as a dominant gene. These are few examples of tumor suppressor genes and oncogenes. I'll give you a couple of seconds to look at it, or you can pause the video and look at it at your own time. Now, this is how the cell would grow in a normal tissue. The cells would grow, migrate, and the dead cells would shed out from the outer layer. Suppose the cell has a problem. It starts with a small single cell which becomes abnormal, and that cell is called as hyperplastic. That means the cell starts to become uncontrollably growing in a fashion where it cannot stop its growth. So it is called as hyperplasia. Then it moves on from that state to a condition where the cells start moving from the localized position and gets displaced, but still it is in a local position, so it is called as dysplasia. Once the cell starts growing abnormally or becomes a clonal expansion of that cell and becomes too much, which is called as carcinoma in situ. So now the cancerous cells are localized in a particular location only, but these cells can enter the bloodstream and travel to different parts of the body and then start localizing in different parts of the body. That is called as metastasis. So these are few examples of things which can cause cancer in our body. So most of the things which cause cancer are environmental. 5 to 10% are linked to the genes that we have in our body which can become abnormal, but most cancers are induced from our lifestyle. Could be from the radiation or from the chemicals that we inhale, probably from cigarette smoke, or from the food that we take, or some kind of bacteria, Helicobacter pylori, which was the first bacteria which was associated to the formation of cancer. Then you have other viruses like human papillomavirus and various other forms which could cause cancer. And to the left side of the screen I have shown you some foods which can cause cancer. The top ones, the smoked foods and processed foods, are definitely agents which can cause carcinogenesis. The ones that are down on the right side of the screen shows green leafy vegetables which can help you improve your health and keep you from getting cancer. Thank you for listening to this topic.